Welcome to today's webinar titled A Masterclass on Research Reporting and Ethical Reproducibility in Life Sciences, conducted in collaboration with the Future Science Group, an independent publisher focused on breakthrough research in medical, biotechnological, and scientific research. This advanced session will help to guide researchers through the underlying causes of poor data reproducibility and ways to circumvent these pitfalls in their data. Today, we have with us Dr. Kayla Chase and Miss Francesca Lake, who will discuss data reproducibility, an issue currently plaguing biomedical research. Dr. Chase is a published author and has worked for more than a decade in the dynamic field of neuroscience, psychiatry, and molecular biology research. She completed her PhD in neuroscience from the University of Illinois at Chicago with a research focus in psychiatry. Currently, she is associated with the School of Art Institute where she teaches bioethics. Dr. Chase is also the recipient of the NARSAD Young Investigator Award and NIH Loan Repayment Award. In today's session, Dr. Chase will discuss the factors contributing to the lack of reproducibility and methods to address low reproducibility. Ms. Lake joined the Future Science Group following a master's degree in biology from the University of Nottingham and is currently the head of Open Access and editor-in-chief of Biotechniques, the International Journal of Life Science Methods. As the latter, she is involved with helping biotechniques authors report new methods that are ethical, robust, and reproducible. In this session, Ms. Lake will discuss common reproducibility pitfalls in life sciences. We welcome and thank you both for agreeing to do this webinar today. Before I hand over the mic to Dr. Chase, I'd like to give you all a brief introduction about Enago. We are the world's leading author services provider, and our author first, quality first approach ensures that our authors receive only the best quality output and excellent user experience at all times. This unrelenting focus on authors' needs means that we never compromise on our quality systems and processes at any cost. All our services are designed keeping in mind the author's needs. Enago Academy is our author training platform that educates authors through several resources, including webinars as well as live workshop sessions. We also have an e-learning platform for researchers called as Enago Learn, along with a live one-on-one consultation platform called Enago Consult. Enago offers world-class services to researchers at different stages of their research career, such as two-editor system English language editing service, academic and non-academic translation service, manuscript preparation service, and publication support service. The Enago promise guarantees that a manuscript edited by us will never be rejected based on poor language. On a rare occasion, if rejection occurs, we will take full responsibility for re-editing the paper for resubmission at no additional cost. Enago's comprehensive after-sales services will support you at every step of the way until your manuscript is published. They are designed to take care of every need from the time you receive our main services until you are published. Enago's research communication services are designed to help you effectively present your research data. In case you are interested in any of these services, please feel free to email to us at academy@enago.com. Now, without any further delay, I hand over the mic to Dr. Chase.
Great, thank you. So thank you so much for joining us. I'm really thankful for the opportunity, and I'm really happy to be here with you guys today. So what we're going to talk about is we're going to focus on, an overview of the replication crisis, some factors contributing to low reproducibility in science, some methods for addressing low reproducibility that you can implement within your laboratory, and then research reporting of ethical considerations by the future research group, which will be Francesca. So let's begin by discussing the replication crisis that science is currently facing. So in theory, researchers should be able to recreate experiments, generate the same results, and arrive at the same conclusions, thus helping to validate and strengthen original work. However, too often, scientific findings in biomedical research cannot be reproduced. In 2016, the publisher Nature asked around 1,500 scientists for their thoughts on reproducibility. Over 70% said they tried and failed to replicate another group's experiments. In anonymous surveys, an average to 1 to 2% of scientists have admitted to fabricating or falsifying data at least once. This population is actually not of interest in this talk. But what is of interest is that in those same surveys, almost 34% admitted to questionable research practices. Some of those questionable research practices we will discuss today and methods to prevent them. So the replication crisis is an ongoing methodological crisis in which it has been found that many scientific studies are difficult or impossible to replicate or reproduce. The replication crisis affects social sciences and medicine most severely. The inability to replicate the studies of others has potentially grave consequences for many fields of science in which significant theories are grounded on unreproducible experimental work. Failing to replicate a study and a finding does not actually make the original finding wrong, but it does leave us in a precarious situation. An inability to come to the same conclusion also doesn't invalidate the data itself. Context is everything, and a different conclusion might not make the data or even some of the findings bad. So let's briefly touch on some of the factors contributing to the lack of reproducibility in science. There's a lack of access to methodological details, raw data, and research materials. There's inconsistencies in managing and analyzing data sets. There's poor research practices and experimental design. There's selective reporting and data analysis, including cherry picking. There's researcher bias. And there's also a competitive culture in research that rewards novel findings and undervalues negative results. So let's start by talking about some methods you can use and incorporate within your own laboratory to address low reproducibility. So some of these topics we'll go further into detail include pre-registration of scientific studies and pre-hypothesis reporting, sample size estimation, improving your statistical reporting, standardization reporting of methods and results, sharing of your raw data, and publishing null results and replication studies. So pre-registration of scientific studies and pre-hypothesis reporting is when authors submit a description of the study hypotheses, methods, and analyses before data collection. This allows the researcher to elucidate the goals of their study and the primary outcomes. Pre-registration does not require every detail to be specified in advance. Researchers can leave some methodological and analytical details to be decided as the project goes along. Also, researchers can deviate from the pre-registration plan whenever they judged it appropriate. In such cases, the researcher would explain in the manuscript why the unplanned deviation was deemed appropriate. This actually builds on initiatives such as pre-registration of clinical trials, which is required by law in the United States. There are several impacts of pre-registration on reproducibility in science. First, it prevents data dredging for positive results, as all hypotheses, statistical analyses, and study populations are defined beforehand. It also encourages the publication of studies with rigorous methods, allowing others to assess the validity of methods before data collection. It also limits publication of only significant findings, which could lead to an implementation of questionable research practices. So one research study examined 296 discrete pre-registered hypotheses and found that overall, 61% of the pre-registered hypotheses were not supported by the results of those studies later published. So how does one pre-register? There's several. Some of the better known places for pre-registration are the Open Science Framework, the Center for Open Science, Evidence in Governance and Politics. There's also clinicaltrials.gov, as I mentioned, the AEA registry. There's a website called As Predicted. There's trial registries in the WHO, the WHO registry network. There's also select journals that support pre-registration. There are around currently about 140 journals using this format. Such examples include Psychological Science and Nature Human Behavior. If pre-registration is through a journal, the plan is vetted through peer review, and the publication is provisionally guaranteed based on the author's follow-through with the proposed protocol. Pre-registration, in its simplest form, is a one-page document answering basic questions such as, what question will be answered and studied? What's the hypothesis? What data will be collected, and how will the data be analyzed? In its most rigorous form, a registered report, researchers write almost an entire manuscript, minus the results and discussion, and submit it for peer review at a journal, which decides to either accept it in principle. After the work is complete, reviewers simply check whether the researchers stuck to their own initial protocol, and if so, the paper is published, regardless of what the data show. Another method is sample size estimation. So before doing an experiment, it is critically important to calculate the statistical power to estimate sample size needed to detect an effect of a certain size with a specific degree of confidence. Underpowered studies are extremely common. Statistical power analysis is critical in experimental design. Underpowered studies do not provide accurate insight. Sample size for any study depends on the acceptable level of significance, and that's the p-value of your choosing, generally 0.05. The power of the study, which is the probability of failing to detect a difference when there actually is a difference. The expected effect size, so for example, a 10% improvement in a therapy. The underlying event rate in the population, and this can be estimated from previously reported studies. And the standard deviation within the population. So that's the dispersion or variability within the data. There are several impacts of sample size estimation and reproducibility of science. It boosts the validity of studies by minimizing type 1 and type 2 errors. And it increases the level of security that you want to reject the null hypothesis. It also removes the chance of researcher bias to manipulate sample size to achieve statistical significance. There's a great free program online called GPower, which will actually help you calculate your statistical power analyses. So another great way to improve reliability in your science is through improving statistical reporting. So this is a multi-pronged approach requiring various mitigation techniques. So first, automate your data analysis. Automation is a practice of using computer systems and processes to perform analytical tasks with little to no human intervention. Data analysis usually requires data acquisition, quality check, cleanup, exploratory analyses, and hypothesis-driven analyses. Pipelines can automate these steps. They process raw data in a suitable format and analyze it with statistical tools or machine learning models in a streamlined way. In addition, automating certain parts of data analyses save time. There's also data analysis, open source script, and code sharing. So we often write new computer programs to solve specific problems over the course of our research. Some of these programs may be relatively small scripts that help analyze our data, and these rarely get described within papers. As authors, we can maximize the chances that other scientists can reproduce our computations find errors, or reuse our methods by sharing these. In addition, open source programs can be scrutinized and improved. I realize that it may not be possible or practical to share all code and data needed to allow others to reproduce your work, but key items should be shared, such as novel algorithms or analyses. Ideally, you should share enough code, data, and documentation to allow at least one key figure of your manuscript to be reproduced. There's some other great resources for open source script and code sharing, one of which is the Jupyter Notebook, but more commonly used is Git and GitHub. There's also a discussion around changing statistical significance. One way is to reduce the criteria for what's known as statistically significant from p less than 0.05 or 1.10 to 0.01 or 0.005. Some potential objections to this include that the false negative rate would become unacceptably high. However, failing to reject the null hypothesis is not the same as accepting the null hypothesis. Fundamentally, not addressing changing the statistical significance criteria fundamentally does not address questionable research practices such as multiple hypothesis testing, p-hacking, publication bias, etc. And also perhaps the threshold should be different for different research communities. Additionally, you can improve your statistical reporting. So many published papers contain statistical errors, making findings unreproducible. Many of these errors seem to be caused by simple typographical mistakes. Some tools to improve this will be the free tool StatCheck25. So this program extracts statistics reported in APA citation style and checks whether the reported p-values match the reported test statistics and degrees of freedom. Similarly, GRIM26 tests can be used to check if reported means are compatible with sample size and number of items. Some of the impacts of improved statistical reporting on reproducibility in science are that it mitigates research analysis bias, especially when you automate your data analysis. It also increases transparency and replicability of data and analysis. It addresses inconsistencies in managing and analyzing data sets, but it also minimizes the rates of false positives and increases your data validity. So a critically important way to increase reproducibility in science is through standardized reporting of methods and results. To help ensure that essential research information needed to assess quality is included in journal articles, it is important that research methodology is thoroughly described to help improve reproducibility. These are standardized guidelines intended to provide authors and reviewers with recommendations for the reporting of research design, data, and analysis, with a heavy focus on methods and results sections. There exist different guidelines for different study types, but in general, researchers should report key experimental parameters, such as whether experimenters were blind, which standards and instruments were used, how many replicates were made, how the results were interpreted, how the statistical analyses were performed, how randomization was done, and what criteria was used to include or exclude any data. The origin of these guidelines can be from publishing journals such as Nature and PLOS, or independent organizations. There exist many different standardized reporting guidelines, and I'm not going to list them all, but I will go into a few that focus on preclinical data. So of note, standardized reporting guidelines that we utilize constantly and we don't think about are the APA and the AMA, so the APA, American Psychological Association, and the AMA, American Medical Association citation guides. So I'm going to start with the umbrella, or with the Equator Network. So the Equator Network is an umbrella organization that, quote, unites researchers, medical journal editors, peer reviewers, developers of reporting guidelines, research funding bodies, and other collaborators with a mutual interest in improving quality of research publications and of research itself. The Equator mission is to achieve accurate, complete, and transparent reporting of all health research studies to support research producibility and usefulness. Their goals, while perhaps a bit more in depth than this, ultimately are to maintain, develop, and promote the use of online resources related to health research reporting. This project began in 2006 in the UK with a goal to map the current status of all activities aimed at preparing and disseminating guidelines on reporting health research studies, identify key individuals working in the area, and establish relationships with potential key stakeholders. Since then, the network has expanded worldwide. There's the UK Equator Center, the French Equator Center, the Canadian Equator Center, and the Australia-Asian Equator Center. Under this umbrella, there exist reporting guidelines for several main study types, and here is the list that they have. Let's begin at the top of the list with CONSORT. So CONSORT stands for the Consolidated Standards of Reporting Trials. It's an evidence-based minimum set of recommendations for reporting randomized trials. This is clinical trials and experimental studies. It offers a standard way for authors to prepare reports of trial findings, facilitating their complete and transparent reporting, and aiding their critical appraisal and interpretation. It consists of a 25-item checklist and a flow diagram that focuses on reporting how the trial was designed, analyzed, and interpreted. The flow diagram displays the process of all participants through the trial. Several extensions have been developed to give additional guidance for specific designs, data, and interventions. The checklist gives guidelines on even the title, the abstract, methods, results, discussions, and other information, including funding sources. The beginning of the checklist is shown here. The CARE is the reporting guidelines for case reports, again under that Equator umbrella. It contains several resources of clinical case reports, including a flow diagram for how a clinician should consider approaching collecting data when seeing a patient or doing a chart review so that it can be systemically collected and written with CARE guidelines. It also includes a checklist of information to include when writing a case report. Again, the beginning of the checklist is shown here. I want to note that the checklists I'm showing are all available for free on the Equator website. The ARRIVE checklist is for animal research reporting of in vivo experiments. The guidelines are intended to improve the reporting of research using animals, maximizing the information published and minimizing unnecessary studies. On their website, you can download a checklist designed to be used when submitting manuscripts describing animal research, an example again of which is shown here. The guidelines are translated into several languages: Chinese, French, Italian, Japanese, Korean, Portuguese, and Spanish. They offer guidelines for all sections of the manuscript, again, starting from the title, the abstract, the introduction, methods, results, and discussion. Some additional guidelines not found on the Equator Network include EASE, so the European Association of Science Editors. Their goal is to improve the global standard and quality of science editing and to provide simple, clear advice aimed at taking international scientific communications more effective. They have a toolkit for both authors and editors, and their toolkit includes a concise guidance for structuring and writing manuscripts, which is updated annually. It includes appendices delving deeper into correctly writing abstracts, paper cohesion, ethics, language plurals, writing simplicity, spelling, and text tables. Another method to increase reproducibility within your lab is the sharing of raw data. So as a researcher, you are increasingly encouraged, or sometimes even mandated, to make your research data available, accessible, discoverable, and usable. Some examples of information that can be shared publicly are your raw or processed data files, metadata, software, code and dependencies needed to run that code, models, protocols, and methods. Once the data is collected, it can be released in a variety of ways, from closed networks within an organization to platform-dependent sharing between peer organizations, publishing with closed licenses, and publishing with open licenses. The impact of raw data on reproducibility in science includes a greater transparency which may boost public faith in research. It also improves access to research for those outside health and academia. It reduces the need for funding of repeat work. It accelerates scientific discoveries and enables scientists to collaborate at a meaningful level. And it reduces the likelihood of researcher bias to select only results that support a prevailing attitude or only confirms previous work. When raw data sharing, select the repository that is most appropriate for your data type and discipline. Some examples include Dataverse, Dryad, Mendeley Data, the Open Science Framework. I do want to say that please be aware of any personally identifiable information when data is published. Data can have real consequences for real people, often harmful consequences. It is very important to try and anonymize information before publishing in any way at all. So every other suggestion has been at the level of the individual researcher, but there are two more systemic level changes. The publishing of null results is fairly uncommon. In an analysis of nearly 30,000 presentations made at scientific conferences, fewer than half were ultimately published in peer-reviewed journals, and negative or null findings were even less likely to be published than positive results. In addition, clinical trials with positive findings are published more often and sooner than negative or null results. This is a great disservice to science, as some null results represent potentially important discoveries. At the very least, leaving null results unpublished results in research conducting is wasting time and resources. Many researchers aren't motivated to publish null findings because the effort required to do so may not ultimately be rewarded by acceptance of the research into a scientific journal. However, there are journals that publish exclusively null findings. There's the Journal of Articles in Support of the Null Hypothesis. It's a prime example. It's existed since 2002 and publishes twice a year. PLOS One also has a collection, Missing Pieces, Negative, Null, and Inconclusive Results. Some journals will publish a null results in brief. Prime example of this is the Journal of Psychopharmacology. Another systemic change that is needed is the funding and publication of replication studies. A replication study is the repetition of an original study to evaluate, reconfirm, or extend the findings with different situations or subjects to determine if the basic findings can be applied to new circumstances. A literature review of 891 papers found that only 3% of papers in the subfield of human-computer interaction had even attempted replication. While critically important to scientific knowledge, there is a very real bias for publication of novel results of not replicated studies. Inago Academy, who's actually hosting this webinar, has published a great article on why journals don't like replication studies. I will include a link to this within the chat. They've also published an interesting article on the clear example of the publisher Nature refusing to publish replication attempts of research. Given the level of importance some institutions place on publishing in high-impact journals like Nature to getting a job or tenure, this can result in a clear statement discouraging replication studies. I'll also post that link in the chat. There are ways to publish replication studies. Most of the replication studies successfully published are of controversial findings or of highly cited works. Additionally, embedding replication studies and results within other sufficiently novel studies is another way to publish these findings. There also remains an additional question of how many replication studies are enough. 1, 2, 5? A final blow to reproducibility studies is funding. The lack of explicit grant funding to perform replication studies significantly limits the initiation of these studies. However, the impact of systemic changes on reproducibility in science is a mitigation of a culture that only rewards novel and statistically significant findings, leading to selective reporting and researcher bias. So in summary, some of the things that we can do within our laboratory: pre-registration of scientific studies and pre-hypothesis reporting, sample size estimation, improving statistical reporting, standardized reporting of methods and results, sharing of raw data, and then publishing of null results and replication studies. So I want to leave you with a real-world example of how working to address low reproducibility in science through standardized methods and sharing of raw data can have serendipitous and positive outcomes. In 2017, Gordon Lithgow, Monica Driscoll, and Patrick Phillips published a comment in Nature. In this, they discussed the impacts of learning that labs could not replicate a groundbreaking finding of a molecule extending the lifespan of C. elegans, which is a tiny round worm. It's about 1 millimeter long. C. elegans are a very common model organism used in genetic and developmental studies. Due to this and several other aging studies that were having issues with replicability, the National Institute of Aging at NIH stepped up and instituted an intervention testing program and tasked 3 researchers with different areas of expertise who had not worked together before to systematically test aging interventions on C. elegans. Their first task was to establish a standard protocol across all labs. But from the first discussion, they realized the variation between the labs was endless, ranging from how one physically transfers the C. elegans from one agar plate to another. This could impact lifespan by a day, to simply deciding how to determine age, when the C. elegans hatched, when the C. elegans eggs were laid. Ultimately, once these differences were ironed out, a standard protocol was decided, and variability between the labs decreased significantly. However, even after the standardization, they found some significant variation within the trials. After dozens of experiments, they realized that some cohorts of worms would separate into one of 2 models of aging, short-lived or long-lived. They're now working together to search for molecular differences that distinguish short-lived and long-lived batches of worms within the same strain, a phenomenon they could have never uncovered had they not eliminated nearly all other sources of variability. So with that, I'll turn the presentation over to Francesca Lake. She's the head of Open Access and editor-in-chief of Biotechniques. Thank you so much.
Hello everyone, thank you for attending today, and thank you Dr. Chase. That was a really good overview of the problems and some of the solutions. So for those of you who are unfamiliar with Biotechniques, I'll just give you a short introduction. Biotechniques is an international journal publishing primarily methods articles, be they new methods, updates to existing methods, or new tools, etc., all for use in the life science laboratory. We also have a news website where we publish the latest news of interest to the life science community, and interviews and opinion pieces from key experts discussing important topics such as reproducibility. The journal launched in 1983, and it is both peer-reviewed and supported by an international editorial board. So here I'm going to go through some of the common issues in research and its reporting that we as a life science journal see or are involved in the solutions for. These include concepts such as the availability of data, software and methodology, be that to a reuser or a peer reviewer needing to assess the work, resource and reagent issues, and the measurement of reproducibility, that is knowing whether you can trust that a method will work for you. I'll also touch on some of the initiatives that exist that are seeking to help improve reproducibility, and also the lack of publication of confirmatory results, which Dr. Chase already mentioned. Okay, so first of all, we'll start with how can we measure the reproducibility of an experiment. So reproducing an experiment for reproducibility's own sake is expensive. One example of this is another independent validation and verification project that was undertaken recently at DARPA, which where reproduction studies have been performed in conjunction with the original studies. These require between 3 and 8% of the original project's research budget, with one particularly complicated one requiring the purchase of equipment at 200,000 USD in order to recreate it. It shows you just how expensive they can be. What's more, as Dr. Chase mentioned, confirmatory studies often struggle to get published owing to the industry's focus on impact, meaning they generally quite often don't get done. The DARPA project goes some way to try and ameliorate this. The validation studies performed there are intended to be published as supplemental material. Hopefully that's a concept that will be utilised quite a lot in the future. Other initiatives seeking to help measure reproducibility include CITE, which is spelled SCITE_, interestingly, which uses deep learning to classify citations to whether they're in agreement, neutral, or negative about the original study. This can then be included as a metric on the original manuscript. So you can see an example from Biotechniques on the screen where 15 citations have been marked as in agreement with the original. The majority are neutral. They generally will be, as the citing study might simply mention the original briefly. However, the availability of the positives and negatives is quite useful to researchers. There's also some softwares available that allow the sharing of methods, which can then be marked as working or not working by others, and reused and edited and so on. I'll speak to this kind of option more in 1 second. So delving deeper into what can be done, I'll start with supplementary materials. Across the life sciences, it's integral to ensure that data, software and protocols used in or generated by a study are accessible, preferably openly, and remain so to enable that maximum reuse, reproducibility and longevity. At Biotechniques, we point authors to re3data.org, which provides a list of data repositories authors can use to ensure this, and also provides information that can be cited in the manuscript to ensure it can be found really easily. It also means that authors don't have to hunt down the corresponding author's email address to find the information, because as we know, those quite often change. In terms of software, we regularly see authors posting their software programs and so on resources such as GitHub, which again provides a link that can be cited in the research article and ensures reusers can find the information easily. There are also various sites where research methods can be posted in a recipe style format. So Biotechniques used to publish these as a supplementary material in a PDF format. While that was a step forward, these could be hard to find and were uneditable. Once published, the protocol couldn't be updated. So we now recommend authors post their methods on protocols.io, and an example of this can be seen on my screen. Protocols.io provides a DOI, meaning the protocol itself is citable and again can be linked in the main research output. It can also be forked, which means other researchers can copy the protocol into a format that's editable for their own use and allows the preservation of the original. This kind of program is therefore incredibly useful for both the original authors and the readers. It also provides metrics, which you can see here, so authors can see how the method is being used and hopefully see that it's being useful. It provides some copyright protection for the author's peace of mind, information on materials, including catalogue numbers, so readers can ensure they're using the appropriate material. It also provides the opportunity for reusers to ask questions of the authors, which is then sent to them and they can answer them. And it provides versioning. So if the authors find a new update or they find a lot of reusers are having a specific problem, they can come in and just update it. And then that link still remains to the research output. So anyone now using the article can go and see the updated version. Protocols.io also provides nice QR codes, which we use in the print versions of our articles, so that our print readers don't miss out on that information either. Another major issue facing reproducibility in the life sciences is problems with reagents. Cell line authentication and antibody validation are just two of the reagent-based problems that have had quite a large impact on reproducibility. But there are ways of ameliorating the issue, and things are improving in this arena. So I'll just quickly start with cell line authentication, which has been historically quite a large problem. And you can see a snapshot of some of the things that have happened in this area on the screen. A major improvement in this area was the launch of ICLAC, which was set up in 2012 to make cell line misidentification more visible, and currently has a register of misidentified cell lines, I think containing more than 400 known false ones. And things are improving in this area for sure. Many publications and grant applications now require cell line authentication to be done, and organisations exist to help researchers do this accurately. However, there's still quite a large issue. For example, gifting of cell lines still occurs, and those have been shown to have an increased occurrence of cross-contamination, and pirate cell suppliers have appeared. So in order to help with this issue, researchers using cell lines should ensure they're authenticating their cell lines. And peer reviewers should feel able to ask editors to provide information on cell line authentication if it's not available in manuscripts when they're reviewing that data. If the cell line appears in the ICLAC database, cell line authentication data such as short tandem repeat profiling should be provided. And we recommend authors read resources such as Cellosaurus, which is both excellently named and also provides a really nice knowledge resource that attempts to describe all cell lines. It currently contains over 100,000, so that's quite helpful. Okay. So antibody validation is another major issue impacting reproducibility. In one study by the Human Protein Atlas, they examined over 5,000 commercial antibodies and showed that over half of those were unsuitable for the proposed application. You can see in this infographic on the screen that this 2015 survey found that the US was spending $350 million a year on research that had bad antibodies and therefore wasn't necessarily valid. And another study done in 2017 suggested that 2 decades of research had been wasted, as the original paper, everything had then been built upon, had not validated its antibodies, and the results were indeed invalid. As such, you can see this is quite a large waste of time and money for something so small, both for the original researchers and any researchers subsequently trying to use their results. So what can researchers do here? The main thing is being aware of the limitations of antibodies and trying as much as possible to choose antibodies shown to work in the application it's being used for. Review sites exist for when you're purchasing antibodies. Antibodypedia is a really nice one. If possible, researchers should validate their antibodies using checklists. These are now available online I think they're freely available. There's a nice one by Weller in 2018, which is cited in the article on the screen, if anyone's interested. And we also recommend that researchers use RID numbers in their research outputs. So RIDs, Research Resource Identifiers, is an initiative that provides unique identification numbers for biomedical reagents. That includes plasmids, organisms, antibodies, and it also includes software tools and databases. It's essentially similar to an ORCID, but for reagents. So ideally, researchers should be including RIDs when they are citing any of this information in their manuscripts. There's quite a nice website shown on the screen where you can go in and find RID numbers should you require them. And I've also included on there an example of what they look like when they're included in a publication. If RIDs aren't used as a minimal requirement, manufacturer information and the catalogue number should really be provided to ensure subsequent users can source the same material and ensure they're reproducing the work properly. So that's everything I have for you today. So thank you for listening.
Yes, so once again, thank you, Dr. Chase and Ms. Lake, for conducting an insightful session today. Our attendees have indeed gained a lot of critical information. Thank you.